Good morning, friends. Today I am here with Reproduction in Animals, part 2 of 8th standard. In today's topics, we are going to discuss development of embryo, types of asexual reproduction, and cloning. So let us start the session. First, it is about development of embryo. Whenever there is fusion of male and female gamete, then it leads to fertilization. And the fertilized egg, that is zygote, divides and de-divides to form a group of cell which is called as embryo. This embryo get attached to the wall of the uterus and then it develops into different body parts like hands, legs, head, eyes, ears. So this is called as fetus. So fetus is the developing embryo. When the development of fetus is complete, then mother will gives birth to the baby. Animals which give birth to their young one, they are called as viviparous animal. For example, humans, dogs, cow, cat, etc. And some animals lay eggs. They are called as oviparous animals like frog, fish, butterfly, lizard. They lay eggs. So they are called as oviparous animals. Here you can see the stages of development of early embryo. Here it is the fetus in the uterus. Here also you can see the fetus in the uterus. That is, uterus is also called as mother's womb. Now, some animals show different stages in their life cycle. For example, the life cycle of silkworm, butterfly, etc. are first they lay eggs, then egg is converted into larva which is called as caterpillar. Then caterpillar develop into pupa and finally pupa develop into adult. And the life cycle of frog also consists of from egg it will develop into larva which is called as tadpole, and from the tadpole the adult frog is formed. So this type of development is called as indirect development. And the transformation of larva into an adult, it is called as metamorphosis. This figure indicates about the butterfly life cycle. You can see that after laying egg, that egg is converted into larva. Then larva is converted into pupa and finally the pupa is converted into the adult butterfly. Here you can see the life cycle of a frog. First the frog lay eggs. Then that egg is fertilized. Finally fertilization, it will lead to the embryo, then tadpole with 2 legs, then tadpole with 4 legs, and finally this tadpole with 4 legs converted into metamorphosis takes place to form the adult frog. Now let us see types of asexual reproduction. There are different organisms in which asexual reproduction takes place by different methods like budding, binary fission, spore formation, regeneration, etc. But here we are going to discuss only two, that is budding and binary fission. So first we will discuss about budding. So in budding, a small bulge-like structure develops from the body, that is called as bud, and this bud grows and develops into new individual which finally detach from the parent body. For example, it is seen in hydra and yeast. In case of yeast, chain of budding is seen. Second one is binary fission. In this method, the first nucleus divides into 2 nuclei, then there is division of cytoplasm, and finally they form the 2 daughter cells. So this division of nucleus is called as karyokinesis, whereas the division of cytoplasm is called as cytokinesis. So first karyokinesis takes place, then cytokinesis takes place, and finally single individual can give rise to 2 daughter cells which can form the new individual. For example, it is commonly seen in amoeba, paramecium, etc. Here you can see the hydra budding. In the first figure, the bulge is appearing. Now in the second figure, you see that it is growing, and in the third figure, it is already grown, and in the fourth, it get detached from the parent body. Here it is figure of binary fission in amoeba. You can see here the nucleus elongated, and it forms two after the division of nucleus and cytoplasm, and it will leads to the two daughter cell. Now the last topic is cloning. What is cloning? Cloning means carbon copy. So it is the production of exact copy of a cell, any other living part, or a complete organism. So cloning may be a cell, cloning may be other living part, or cloning may be of complete organism. So first of all, cloning of an animal was successfully done by Wilmot and his colleagues at the Roslin Institute in Edinburgh, Scotland. So they successfully cloned a sheep, and they called the sheep as Dolly. Dolly was born on 5th July 1996, and it was the first mammal to be cloned. Here you can see the Dolly with Wilmot. This is the cloning of a sheep, Dolly. This is also figure of Dolly. You can see the exact carbon copy. Thank you. For any kind of doubts, please write your question in comment section.